renal denervation is back in focus because we designed, as investigators and multiple investigators across the Atlantic, new studies using new type of catheters than the initial one. And the fact is, by using these new methodologies for renal denervation, but also new designs of trials, we have been able to show homogeneous results from one trial to another trial. This is very important to get the maximum evidence about the efficacy and safety of such approach. There are different types. You have basically the two types, so the major types we have been investigated is either using radiofrequency ablation. It's a small current of very low intensity and going through the whole artery wall from the endothelium to the adventitia and making this ablation, and it's making a thermal lesions. With ultrasound ablation, there is a difference. That is, you are going not to use radiofrequency, but highly focused ultrasound to induce this thermal lesion. But the probe is inside the cooling balloon, so you prevent any endothelial damage or damage to the media of the artery. And also the difference is that the ultrasound catheter goes mainly inside the main renal arteries, whereas with the other catheter, in terms of making the renal denervation, there are ablation different points, specifically distally, even after the renal artery bifurcation. These two trials, SOLO, which was designed for patient either naive, hypertensive naive with no meds, or of really 0 to 2 antihypertensive meds, and TRIO, which has been conducted in patients with severe unresistant hypertension, had a similar design. So both trials were designed for superiority, and the sample size allowed us to demonstrate superiority. Because of this design, and they were run in parallel, so we did the first pool individual patient analysis. So, and this brought us with around 146 against 139 patients in the denervation group and the sham group. So it gives us much more statistical power by combining in this individual data. The first thing we found is that indeed there was no heterogeneity between the trials, and specifically in terms of the response, blood pressure response, and also the medication burden. So this was the two major endpoints we were analyzing. So in summary, what we showed by combining these two trials is that during 6 months follow-up, from randomization to 6 months, the average drop in blood pressure, either evaluated by ambulatory blood pressure monitoring or home blood pressure monitoring, was around 5 to 6 millimeters of mercury systolic blood pressure difference, which is highly statistically significant, but also highly clinically significant in terms of ambulatory blood pressure, which is larger than the efficacy than office blood pressure drop of 5 to 6 millimeters of mercury. And this occurred with less medication given to the patient randomized the renal denervation group. So this increased indeed the evidence around let's say the midterm efficacy of renal denervation using ultrasound catheter for 6 months. And it was also extremely safe, no safety issue, and with a reduction in the medication burden. At the level of the European Society of Cardiology and European Society of Hypertension, we are going for, we hope, for the next ESC meeting, release the new guidelines for renal denervation, taking into account all this new evidence coming from the different trials. This is an important question where you are asking, and this means that we need to work up to exclude different types of phenotype which will not be should not be treated by renal denervation. So we should basically focus with what we know from the trial. These are younger patients, difficult to control patients, those who are also willing to have renal denervation because they absolutely don't want to take any medication or are completely non-adherent to treatment, or some phenotype where people have as adverse events to any medication you prescribe them. So the alternative is just looking to them and they had persistent high blood pressure and go to an event. So, and we try to find some specific phenotypes where there's activation, overactivation of sympathetic nerve activity, those with sleep apnea, obesity, high blood pressure during nighttime, etc. So high heart rate. So these are features where the responsiveness to renal denervation is larger.